Okay, so good afternoon. We should start. So he's Martí Checa. He's now in Oak Ridge National Laboratory. So Martí has done the PhD with Gabriel Gomilla in IBEC, so doing bio things near physics, but not there. So after he did the postdoc in Oak Ridge already, and now since 2024, he's a staff scientist in Oak Ridge. So he will speak today about this or not?
Yeah.
Okay. About manipulating ferroelectric topologies with the PFM, that I found it very exciting. And so the floor is yours.
Thank you, Ignacy, and thank you all for coming. It's I'm originally from Barcelona, so it's a great pleasure for me to always be at home. And yeah, thank you to the ICMAB and to Ignacy for the invitation. So originally, when Ignacy asked me to talk, I'd send him an abstract, but then he told me it was 45-minute type seminar. So I kind of like, when I was preparing the talk in the plane, I sort of wanted to talk a little bit more big picture. So I'm going to talk about manipulating ferroelectric topologies with an AFM, but I'm going to more give a big picture of what type of things we can do when you achieve a full control of an AFM, of an atomic force microscope, right? And in particular, I will assess a lot the role of the scan path. So we're going to go into detail now with it, but most AFMs scan left to right in the raster scan mode, and we're going to see what we can do when we get control of the piezo's and we start playing with it. So as most of you know, and I actually have some users in the audience, I'm part of a user facility, the CNMS, which is within the Oak Ridge National Laboratory. So we're one of 5 NSRCs, which are national nanoscience research centers. And basically, we operate very similar to a synchrotron, right? There's 2 calls for user proposal, and then the acceptance rate is pretty high, it's around 75%. And then you can either send us samples and we can measure them for you, or you can, come yourself or a student of yours, postdoc, whatever, and do measurements over there. So just one slide about myself, like Ignacy said, for the people that don't know me. I did my PhD from 2016 to 2020 in IBEC under the supervision of Gabriel Gomilla. And I used the AFM, but more for bio-related projects. We used the AFM for electrical characterization of cells. So we were doing the electric constant maps of cells. Apparently the biocomponents, so the nucleic acids and proteins and lipids that form the cells give different dielectric response and have different polarizability. So you can achieve some sort of label-free biomolecular characterization from the dielectric constant signal that the different biocomponents have. Then I was actually the last PhD thesis defended before the COVID. I finished in February 2020. And then I had to go to the US, but I couldn't. Everything got locked. And I did a short postdoc with the group of Gustavo Catalan here at ICN2. And then like 8 or 9 months later, I went to Oak Ridge and I did my postdoc there. Now, still using the AFM, but more applied to material science. A lot of nanoelectrical characterization, PFM, KPFM. I'll be talking about it in a second. And my advisor there was Dr. Liam Collins. And just recently, about a year ago, I got promoted to staff over there, which allows me to, as I said before, work with users, have my own research projects, etc. I consider myself more a microscopist than, the material scientist, in the sense that I'm more like a pilot of a microscope. I know some of ferroelectrics, some of bio, particular cell imaging, and I also have experience in other techniques like finite element simulations and correlative microscopy. But yeah, we can talk about that if you're interested after. this is the outline of my talk. So I'll start with an introduction to microscopy, which is a kind of like a package, which stands for automated experiments in microscopy, where I'm going to show you how we achieve the full control of the AFM software and different parts of it. And then I'll show 3 different applications. One is how can we use the spiral scanning? So instead of scanning left, right, scanning in spiral form to scan faster, right? So do some sort of high-speed AFM, but functional. Then the second part will be on how can we use AFM to write a specific ferroelectric domain configurations. And finally, I'll show some very recent results about some application of writing those ferroelectric domains. So I assume most people here know what an AFM is, but just very briefly, an AFM uses a sharp tip that when brought in close contact or in contact with the sample, is able to feel the local forces between the tip and the sample, right? And so we can reconstruct the topography of the sample. The main advantages for me of an AFM is that you can not only get the topography, but also get some functional information. You can do electrical mapping, magnetic mapping, mechanical mapping, etc. And also you can use it not only in ultra-high vacuum, which it's needed for other types of microscopy with such resolution, but you can work in liquid environments or with controlled humidity, etc. And this is how the commercial AFM softwares look. Basically, most of the times you can do images in different places of your sample, and you can do force spectroscopy, right? So you can apply force distance curves and record the interaction between the tip and the sample. And microscopy, which is this package that we've been developing at Oak Ridge, basically is a library of different Python functions, right, that control an FPGA. This is kind of working, but control an FPGA that is kind of like hacking the AFM controller, right? So we can apply biases to the piezos, we can apply biases to the tip, we can basically achieve full control of the AFM by hacking it through using an FPGA that's controlled via Python. Some examples of what we can do is, as I said, tuning the scan path. So not only left, right, but doing a spiral or doing like a Lisa-Ju type scan, doing a flower scan like this one. So I'll be showing some examples of that. But we can also do different bias spectroscopies, so apply pulses to the tip, apply different waveforms. And not only that, but we can also find specific areas of interest. In this case, this is a PFM image of a ferroelectric domain. That is segmented. I don't know if this will be working. It's segmented as we go, as we take the image. 1 second. Oh. Well, this is kind of similar. This is another example, but we do a lot of work with ferroelectrics, and in this example, we have 2 ferroelectric domains, and we're studying domain wall dynamics. So we take a PFM image, we segment it and find where the domain wall is, and then we can go to specific locations within this domain wall and apply different bias pulses of different lengths and different amplitudes, so we can see how this domain wall moves around, right? Another example, also for the ferroelectrics case, would be to study, how the pulse duration and the bias amplitude of a PFM type experiment works to nucleate a certain domain. So we can, generate an array of different pulse durations and bias amplitudes and generate this type of maps that you can see here of how the domain nucleates and, what are the switching mechanisms that we have under the tip. And another example, which is basically what I'll be talking during my particular applications, are to customize a scan trajectory. So as I said again, we can use that to scan faster. So we now can achieve some dynamic process mapping, right? Correct for drift or migration within the sample. But not only that, we can also use it to scan smarter in a way. So to achieve some sort of effective electric field in a specific direction, or for micro manipulation, additive manufacturing, etc. And all that is needed to, if you want to automatize and you want to start, applying AI or ML to segment images and to apply, reduce the human time in the microscope and apply specific type of routines to your samples. So let's go to the second part. In here, as I said, I'm going to show you how we can use the spiral scanning, and I'll focus on spiral, but we can do it really with any trajectory for faster functional imaging. So I guess most of you are familiar with KPFM. It's an AFM-based technique that applies an AC bias between the tip and the conductive sample. And by nullifying the capacitive forces that appear, you can map the contact potential difference between the tip and your sample. Right? Then this is an example of a 2D material map of its CPD. However, this type of images in most of commercial systems take around 2 to 10 minutes per image, depending on the pixel resolution, etc., that you want. So what happens with dynamic process, right? What happens when you want to have time resolution during a KPFM experiment? So basically, there's two ways of going about that. One is called time resolved KPFM, which would be kind of like a spectroscopic way of doing KPFM, where you sit in a special point and then you apply, you trigger specific process and you map the CPD in that particular pixel. Then you move to the next pixel and do it, kind of in a force map way, which, it's good because it has a lot of temporal resolution down to the nanoseconds in some pump probe techniques that are being developed recently. But it's only good for quasi-static or reversible processes, right? What happens when you have a solid-liquid interface? What happens when you have a battery? Those methods cannot work in there because those processes are normally not reversible, right? So another alternative is to do high-speed KPFM, which would be just basically scan faster. This would be, good for, in particular, in principle for any process, but it's much slower, right? So we did a little bit of research, and the fastest KPFM image that we found out there was this one by Garrett that took 16 seconds to do one frame in imaging mode, right? So here I show you how we developed this spiral scanning. As I said, we achieve external control of the XY piezos that move the tip or the sample around. And then we can design a trajectory, in this case, a spiral. Gives us a lot of flexibility of parameters. How sparse do we do this scan? How dense do we do it? Which dimensions do we use? And then we just, instead of scanning left to right, we scan in this spiral form, right? And the second part of it is, we don't scan all the pixels, so we have to sort of in-paint or reconstruct the pixels that we don't scan. And for that, there's different techniques on the computer vision side of things that allow us to sort of reconstruct the pixels that we don't scan. Then this is always a trade-off, right? Like you can go faster and scan less pixels. But then you'll have less spatial resolution because at the end of the day, whatever you're in-painting, you're not really scanning. It's some sort of interpolation. You can always, what I recommend for this type of techniques is do one image the old-fashioned way. You see what type of features you have in your image. And then if you want to scan a process, a dynamic process where things are moving and changing, then this is a good alternative for that. Then this is the same, 2D flake, and this is an example of, doing a spiral with different cycles. So I can show you the flexibility that we have with this type of scans. And one can see that only with 6 cycles, the algorithm does a pretty good job at, knowing the shape of the 2D flake, knowing that the quantitative values of the CPD, and this would be equivalent to just doing 3 lines maybe on a left to right raster scan. And then in terms of speed, for the speed, I like to show this example where, you know, I took a PZT sample and I wrote some checkerboard-like domain structure. And so I started kind of pushing the limits of the piezos as we drive it. And I could get, you know, 3.3 frames per second, where we still have, you know, a pretty good idea of the underlying polarization structure of our sample. So this is 2 orders of magnitude faster than this 16 seconds per image that we found. Obviously, this will depend on what polarization pattern you have your mapping. You know, if it's very intricate and has a lot of, you know, minute things that you want to map, this gets a little more tricky. But it's you know, it gives us a lot more temporal resolution in the imaging mode. Then an example of a system where we, you know, map some dynamical process while we're doing this spiral scan KPFM. So this is TiO₂ that has different grains. And the experiment here works as following. So we approach the sample, we apply a DC bias for maybe, I don't know, 10 seconds, something like that. So we inject a lot of charges. And then as soon as the bias stops, the tip lifts and starts mapping in spiral scan mode, right? So you get the topography and you get the CPD image. And it looks something like that. So the area in the middle, you know, we can see that we've injected some charges, and we can see the relaxation and diffusion of those charges. In this case, we injected charges in 2 different grains because the pulse amplitude was 10 volts. But then, you know, we can also, you know, do a pulse amplitude of 8 volts, and then we can see that we only inject 1, the central sort of grain. So we can study, you know, the charge diffusion and reorganization with, you know, much faster temporal resolution, which in state-of-the-art KPFM, those processes will be masked because they're too fast to, you know, they happen in the range of seconds or something like that. Then you can do that as a function of temperature, right? So in this case, we did using the same sample, different temperatures, and we calculated, you know, the diffusion constant, supposing that we have that it is an Arrhenius-like process, and we could obtain some activation energy for those charge diffusion within our sample. So as a kind of summary of this first part, I've shown you that with sparse sampling and image reconstruction, we can achieve some sort of high-speed functional imaging with hundreds of millisecond temporal resolution that allow us to, you know, study charge dynamics within, you know, the hundreds of milliseconds to tens of seconds regime. And this is applicable to any AFM mode. So I showed here an example with KPFM, but this is an example for PFM where we had some ferroelectric and not ferroelectric domains, and I ramped up the temperature real fast so that, you know, I went through the Curie temperature of the material, and we see how the, you know, domains disappear in the PFM channel. And when I start cooling the sample again, you know, the PFM contrast appears back. Then this allows us to play with, you know, rates of heating your sample that are, you know, not reachable if you need to, you know, do a full PFM scan after the change in the temperature. So I'm going to go to the second application for this smart scanning. The first part would be how to read, right? How to read faster. I showed in KPFM, but as I said, we've done it with topography, we've done it with PFM, etc. Now I'm going to be showing you a little bit how we can use that to write information in our materials, right? So I don't think I have to introduce ferroelectrics probably to this audience because there's a lot of people know a lot more than me on ferroelectrics. But in case there's someone lost, ferroelectric material is a material that has, you know, remnant polarization, even when, you know, in absence of an externally applied field. And this polarization can normally be, you know, reversed with the application of an external electric field. And when we look at the microscopic dipoles that form this ferroelectric, under certain exotic boundary conditions, one can achieve this weird arrangement, of these microscopic dipoles that are called topological structures in ferroelectrics. So we have a zoology of, flux-closures and vortexes and skyrmions and merons and you name it. So those types of ferroelectric structures or polar structures are real interesting from the application side of things, right? But they're also very interesting from a fundamental physics standpoint because they allow us to probe the energy landscape of this type of materials. Then where do we find this topological arrangement of domains? Well, you can find them kind of serendipitously, like in this manganites from the group of Dennis Meyer. But there's a lot of work done in the last decade or more on strain engineering and chemical engineering during the growth of the material to kind of foster the conditions that allow those structures to sort of appear during the growth of the material. They've also been shown to, appear under mechanical current pressure, or even, geometric confinement, like these nano islands, that form these center-convergent and center-divergent type domains. And very recently, also people have used, weirdly patterned electrodes to generate in-plane electric fields, like this one here, so that you can tune the properties. I think in this case is bismuth ferrite with this, in-plane effective electric field that you get with this electrodes. But the question that I wanted to answer here was, can we use an AFM tip to write, read, and erase those type of domain arrangements on demand and without the need of depositing electrodes and doing kind of complex treatment of the ferroelectric surface? So I'm going to introduce the system that I've worked with for this part, with this lead strontium titanate. But before that, I want to just briefly introduce PFM. So most of you probably know PFM is an in-contact technique where the AFM tip is brought into contact with the sample, and the deflection of this AFM tip when we have a piezoelectric sample that under a bias application deforms in certain ways is measured. So that way we can sort of map the local polarization. Is a technique that tends to be very artifacty because the tip oscillates not only because there is a deformation locally, but because of a million other things. You can have electrochemistry, you can have just capacitive coupling from the cantilever, etc. So it's not the solution to everything, but recently we purchased this interferometric PFM, which basically uses, instead of an optical beam deflection to track the movement of the cantilever, it uses an interferometer, right? So this has a lot less noise, but also it's quantitative, right? So you can know exactly how many nanometers or picometers you're moving your tip up and down. Not only that, but if you do a little bit of trigonometry here, you can reconstruct the 3D sort of vector polarization by placing the interferometer at different positions along the cantilever. So if you do a PFM image with the interferometer at this position or at this position, and then you withdraw it with some mathematical operations, you can read the paper here, then you get the X component of the polarization. Then you do the same with left and right instead of top and bottom instead of left and right. Here you get the Y, and then by averaging the four you get the Z component. So this is good because normally, classically, this has been done through rotating the sample and finding the same spot again. So this kind of allows us to do that without moving the tip and in a quantitative manner. So let's go back to the material system that I'll be showing you here. This is lead strontium titanate. The samples are from the group of Lane Martin. And with this concentration of strontium, the polarization here is in plane. So we have what are normally called superdomains, which are these arrays of A1, A2 domains that have the polarization in plane. And there's 4 possible variants of these superdomains that go along the diagonal of this 100, 010 crystal structure of the material. So basically, in the pristine case, which is what this lateral PFM image is showing, you have the 4 possible polarization directions. Then the group of Beatriz Noeda and others have studied how can we use an AFM tip to switch the polarization state from one of those superdomains to another, right? And how they do it is by using the slow direction of scan as a driving force for that switching mechanism to happen. So basically, by scanning from the bottom to the top with positive bias, you get the same as if you scan from the top to the bottom with negative bias. So you have some sort of trailing electric field that allows you to switch the polarization in plane within those 4 possible states of your sample. Then we wanted to study what is the effect of the scan trajectory on the final domain, and can we use it to sort of generate these topological structures in these type of materials. So we use, again, microscopy. We've also kind of leveraged the use of LLMs, and I'll show a video at the end to sort of help us design the programming part of this. I talked before about how we've used those two for noble, faster scanning modes. So I'm going to be showing now a bunch of experiments that are all done the same way. So I thought it'd be nice to explain first how we do these experiments, because they're all kind of the same. So we start with a pristine sample with all those 4 different polarization states. And we bring the tip in contact, and we scan the old way, left to right, sort of to write a blank slate using this raster scan trajectory, right? So that then all the polarization is aligned into one of these 4 possible directions. Then the step 2 is to use the tip, same tip, but now, with external control. And I'm going to be focusing again on spiral scans, but you can use any scan path that you want. You bring it in contact, you apply a DC bias while you're doing this scanning spiral. And then finally, you can use the same tip to read either lateral PFM or vertical PFM of the domain that you have written. And this is how it looks. So when we apply a negative writing bias in this lead strontium titanate, we see this 4-quadrant structure appear with all the domains kind of pointing inwards, right? Where the blank slate is all pointing in one direction outside, but we see clearly 4 quadrants all pointing kind of towards the center. This is in the lateral case, and then in the vertical case, we see that the boundaries between those quadrants kind of light up, the same as this boundary here, which is particularly where we have the head-to-head and tail-to-tail domain, superdomain boundaries, right? So we hypothesize that what's going on here is that as we generate those charged domain boundaries, the polarization sort of kind of rotates towards the Z direction, right? So we have some sort of center-convergent structure in which those frustrated or highly charged superboundary domains, the polarization goes either up or down. And then what happens when, instead of doing it with a negative bias, you do it with a positive bias? Well, now instead of having a center-convergent structure, you have a center-divergent structure. And you still see that, on the superdomain boundaries, you have, this sort of Z component of the polarization, right? Then we've measured this type of structures with other techniques like polarized SHG and SEM-CL. So that's cathodoluminescence, where we see again those boundaries light up, which tells some information about the defects that we may be inducing in those super domain boundaries. But as I said, we want to go a step further and kind of generate different polarization states. We not only want to scan with a spiral, but we want to design other scan paths that allow us to generate a different polarization structure. So we designed this flower scan, which basically the tip is scanning kind of radially as it spins along this center, and then we see that in this case, and this is highly reproducible, we have, again, 4 quadrants, but they're sort of positioned in this way that it looks like we have some sort of a flux-closure of the overall polarization, right? So we're kind of happy because, the literature says that it's the trailing electric field, so this is slow scan direction, what is driving the switching in this type of materials. And we said, well, when we have a spiral scan, the slow scan direction is the radial direction, right? And this allows us to, engineer center convergent and center divergent type structures. And when we have a flower-like structure, the slow scan is the angular scan. So this generates these sort of flux closure structures. But it's more complicated than that, because when one starts putting the signs in it and everything, in this case, it goes anti-parallel with the trailing electric field. And in the case of a raster scan, it goes parallel with the trailing electric field. So we're doing a lot of phase field simulations to try to figure out what is actually the driving switching mechanism in this type of hierarchical domains. But we think it has to do with an initial nucleation of multiple states and then the dragging of those domains that have been nucleated in the different directions. So this is highly tunable, right? Now we can start writing arrays of these guys. So we can play with the scan direction and generate 4 different states just with positive and negative bias and flower and spiral scan. But not only that, we can also play with sparsity, right? So we can generate these sort of ring-like structures that overall resemble a center convergent, center divergent type structure. We can play with the size of them too. I like this sort of concentric structures, right? Where we do sort of a donut, where the outer part is a center convergent structure, but the inner part is a center divergent. Or, one can think of any, scan path, like this snake-like scan path that aligns the polarization in this weird snake-like domain. And this sort of brings me to the last part of the talk. I don't know how I'm doing in time. Yeah, that's good. Where, I'm going to show you a possible application of this method to write ferroelectric topologies. In this case, we'll do another material. This is bismuth ferrite. The samples are from Professor Jianqi Yang from Taiwan University. And basically, that's the pristine state of the sample. In this case, that's the sort of 3D distribution. In this case, as you know, in BFO, the polarization, there's 8 possible polarization states that go along the diagonal of this crystal structure. And when we do one of those spiral scans, in this case of BFO, what we get is this structure. So the same scan path now produces a completely different polarization structure just because this is a new material and the material switches through a different mechanism. So in this case, I call this a striped closure domain. But yeah, I don't know if that's the correct nomenclature. People can correct me or give me ideas for names later. But I call it striped because each of these quadrants has stripes, and closure domain because if you now put together the 4 polarization directions, you have this sort of stripe closure around the center of these 4 domains. And how can we use this? Well, I'm going to show you that a possible application of those is for multi-state information encoding. Because now that I know how to generate these multi-state domains, I can generate an alphabet. So in this case, it's an alphabet of base 2, where I do a spiral scan in one direction, kind of clockwise and counterclockwise. This generates 2 different stripe closure domains. And now I can start, writing. In this case, I wrote ORNL, which stands for Oak Ridge National Lab, in base 2, right? Using zeros and ones, as in, these 2 closure domains going clockwise and counterclockwise. And this, when you do these structures 1 micron big, more or less, in diameter, this has an information density of, 10 to the minus 7 bit per nanometer square. What happens now if, we engineer a different alphabet, and we can do these 6 different polarization states, which, we go clockwise, counterclockwise, and then we do clockwise on the outside and clockwise on the inside, but applying a negative bias. And you do all the 4 possible, combinations of those. Then you have 6 different characters. And now we can write ORNL, but in base 6, using a lot less space, right? So the information density sort of goes more or less half an order of magnitude higher, right? So when you compare this type of information density with current hard drives where, more or less 1 terabyte per inch square is the information density of those, we're still a couple of orders of magnitude below the current hard drives in terms of information density. But then it becomes a matter of how many of those states can we engineer and how small we can make them to sort of quickly ramp up the, information density of this type of methodology. I'm going to show you how we do this type of experiment. So we have a Python notebook that controls, the scanning engine of the AFM. Then I have an oscilloscope here that, I don't know if you can see from there, but we can track the position of the tip. So it's going left to right, because we always start with making this blank slate that I was telling you. So we have, only 1 polarization state. Then the tip went to the center. We keep going in the Python notebook. And now, the user is going to input which system, which base system we want to use, in this case 6, and which word we want to write. So it's, in this case, ORNL. And then, we keep going with the Jupyter notebook, and the tip we start scanning, will go and start writing directly what the user has, given as an input. It's a slow process, right? This is super slow compared to any, hard drive or anything like that. I like to think that we don't only have to think in terms of speed. So, for example, ferroelectrics have a very high Curie temperature, and they're very good with, radiation and stuff. So maybe it could be good for possible hard drives that need to go to space or need to work in harsh conditions. So, well, this keeps going for a little bit. I don't probably don't want to have you sit here, just watch me play with the AFM. But the last part is kind of to close the full circle. We generate now an array of the 6 different symbols of my alphabet, and we train super vector machine to recognize them automatically. So now we have the full circle where the user can write the information that needs to be stored, measure in PFM using the same microscope, and that PFM image is now given to this classifier, and it gives you back the information that's written there, right? So yeah, that's all from my side. I want to thank everyone that's involved in the SPM development, sample preparation, phase field modeling, and other characterization techniques, and the funding agencies for the support, and you for your attention, and be glad to answer questions.
Stop.
Out of curiosity, I can see that your spiral scans always go from the center out. We try to go from the outside in.
Yes, I have. I haven't played too much, and it's one of the things that I have to start doing. But sometimes you don't even write anything. So again, it depends on the material because we still don't actually understand how the switching mechanism is. But that's one of the things that make us think that, it's some sort of nucleation and then kind of dragging of these boundaries that you generate, right? Yeah.
Question?
You mean during the reading, some sort of artifact because we're reading in spiral, if I'm understanding correctly? So most of the reading that I showed for this part was done in raster scan. We just use the spiral scan for the writing. In this part, we could also implement for the Kelvin images. So over there, it's a little tricky because, as I said, it's a trade-off between the spatial resolution and the temporal resolution. So when you want to, use the spiral scan to actually read faster, and this will take forever, but oh my God, it's too much. Yeah, so this is a comparison, for example, of noise of a normal raster scan image, this one, and the KPFM image. So you see that ours appears to be much less noisy. This is fake because this is just the image reconstruction algorithm acts as a filter and it just reduces the noise by a lot. So actually you still have the same noise floor. So in this we were just using this to say the quantitative values that we obtain are the same. But in terms of noise, you still are gonna have the same sort of noise. It's actually a little better for the piezos itself because the piezos don't like to change directions drastically, and that's one of the things that takes more time during AFM scanning. So actually it's better for the piezos to just go around it like this. It allows them to scan faster, but it's also probably better for them long term. We haven't, all the artifacts are induced more by how you do this reconstruction, not so much by the scanning path, if that makes sense. You can have the different reconstructions. You can have a look at them this is discussed in my GitHub. If you want.
Mauricio?
This memory element that with the last part did you have tried to determine the cable, the variability, length of the interval, this reading time can be an output for?
Yeah, so those are very stable. They stay there for months. I haven't measured them through years, but I'm pretty sure the structure, it's just there. Now, as I said, while I was presenting, this is very slow. So it takes me a couple of seconds to write one of those structures, right? So in terms of scaling, one could think of an array of, tips and then some sort of, continuous scanning with different biases applied and different trajectories. I showed this application as in, we can automate the process a lot and we can control our microscopes very well. However, I do think that this method is very good more from the fundamental physics standpoint of understanding what is the switching mechanism in there, what's going on actually in the energy landscape of this material. The final application is still too early. Yeah.
Question? Philip?
Dynamic processes with picosecond difference?
No. So what I have said is that there's methods that can do KPFM down to the nanosecond. This is more related with pan-probe techniques. It's not standard, just off-the-shelf KPFM. So you normally use 2 excitations, and then the time resolution is given by the difference between the exciting poles and the reading poles. It's not like I've shown here. Like what I was doing here, it's just doing KPFM, but scanning in a different way that allows us to go faster. Now, when you compare these with high-speed AFM, so high-speed AFM has been there since probably 30 years ago already. Toshio Ando. Yeah, this is not good for ultra fast. This is good for this mid-range where you have a process that is not minutes, but it's not nanoseconds, right? Down to the hundreds of milliseconds, we can work there, but not farther than that. Yeah, and the key point here is high-speed AFM has been out there for a long time, and people that do bio have, I don't know if you know this famous actin protein walking in the myosin filament video. I don't know if you've seen this, but they can do images at 5 hertz or like something like that, 50 frames per second, some crazy thing like that. They do video rate basically. However, all the modes that are developed for high-speed AFM are just topography, because the bottleneck for most of the functional AFM modes are related to the functional mode itself. So when we show these in AFM conferences, the high-speed people are like getting 3 frames per second, and 20 years ago we've been doing 50 frames per second. But the key point is that we do something more than topography, which is the advantage of the AFM at the end of the day.
More question?
Great talk, Marty.
Thank you.
So in the second section, you used material light strong titanium, right? Why this material? Usually, I suppose I don't develop technique, but I suppose one develop a new technique, I would choose a very simple system. For example, why this material?
Yeah, well, I could make a story about myself and everything, but I'm just going to be honest. We had that sample in the lab that had nice domains in plane. And I was trying to, I thought that the spiral scanning would be good to tune those in-plane domains. I have tried with other samples that have in-plane polarization too, with more and less success. I think the key in this point is in these samples is that the polarization wants to be in plane, but there's also this sort of hierarchy of the superlattice. So you have a small A1, A2 domains, and the interaction between those is what drives these weird polarization states when you scan in spiral, right? So the answer is because we had it and it had in-plane domains. Now, we've tried others that we also had and they didn't show that nice of a story. And I think it's because most of them don't have that hierarchy of, the superlattice part of it.
This superdomain structure is kind of precondition to apply this technique.
What can you say again?
I mean, is this A1 into this superdomain structure is a necessary condition to apply this technique, or?
Well, in the BFO, you don't necessarily have this A1, A2, because in the BFO, you have some of the 4 diagonals, and you still see some sort of nice structure come out of it, but the switching mechanism is completely different because, the nucleation is different for that material. So it's not necessary, but it plays an important role, I'd say.
More questions? So then I will give couple of questions. So I am still curious. I think that I already asked you this, but when you write let's say the domains that are like this, you see that in the middle, the out of plane signal is brighter, no? And then you say, okay, it is like that. And for the other chirality or whatever, it is negative. But when you write this flux-closure, let's say, you also see this bright and dark image. You know why they tilt out if they do?
Good
so this
question.
is okay.
Good question. So here we have bright because we have head-to-head. Here we have dark because we have tail-to-tail. Then now in the center, we have tail to tail, so we have dark. And then in here, we have, it's hard to see with this color scale, but we have a little bright because the underlying is head and this is head. So it's kind of, then what you're asking me is when you do the flower, right? Here, you don't really have a head, head to tail, and I still see some sort of lighting, right? So I said that we hypothesize that the polarization goes up because I tried to do conductive AFM. I didn't get any current out of it. So one could think that it's charging. And the fact that I didn't have conductive AFM, and I also don't have the data here, but we did phase field modeling, and the phase field showed that if that's the structure that you have, it would relax towards out of plane. So we hypothesized that what we have is on those boundaries out of plane. We didn't do the simulations for this case, because in this case, you have some sort of weird state where it's like, you know, kind of side to head. One could think that in this case, you know, when you zoom in those boundaries, you do have all the cases, because then you start seeing the A1, A2 lattice. And in this lattice, you do have some cases where you have frustration and some that don't. It just probably in this case is less seen, or less, you know, this effect is not as strong as in the other one, because you don't have a direct head-to-head. That makes sense.
Yeah, yeah. And in this phase modeling, but it doesn't take into account that you can have carriers in the lattice, or so obviously it will go up.
We're working on it now for the switching mechanism, because when we try to actually put in the phase field modeling, the tip going around, we don't see the nucleation of this type of domain. So we think that vacancies that you have, you know, the conductivity of your sample, but most of the phase field modeling, it's hard to induce those defects that may be pinning the domain walls in certain spots and stuff, and maybe, you know, the driving force of those type of nucleations. I still don't have a full answer as in why the material switches like that, but yeah, we're working on trying to. Ideally, you know, what I want to get to is some sort of, even if it's just phenomenological, but some sort of model where we have tried this with different crystal structures and different scan trajectories, and can we use that to design states, right? Yeah, that's kind of the next step, I think.
And maybe a little bit related to that, because I have not seen many other labs reproducing similar results, let's say, with this complexity. The paper from Beatriz, it's similar, but the structures are not the same. So maybe related to that. What is the price of the FPGA that you?
The FPGA is not very expensive. It's a normal FPGA with a breakout box. I don't know the price of it, but it's on the tens of thousands. It's not on the hundreds of thousands. Maybe for 30K or something, or 10K, you have one. The key point here is the expertise on controlling this, because I didn't show any of that, but all the FPGA work and the microscopy was developed for scanning transmission electron microscope. So to move the electron beam. And then we just adapted it to an AFM. But there's a lot of work done in Oak Ridge where they move the electron beam in certain directions to, like, you know, get an atom out of the lattice or something. Then the key point here is the expertise on controlling the FPGA, and we have a LabVIEW software that it's like kind of our holy Bible, that, you know, that's where we control with the Python, and that's what allows us to do all this. We've done it with the ciphers, but we've done it with the NanoSurf. Like, as long as you can have access to your controller, it's not AFM limited. We've done it with the Brookers too. Yeah, it's just more about having the expertise of controlling the FPGA and giving the inputs to the AFM controller, if that makes sense.
Okay, so to me it's okay. Question, Marcos.
Thinking it's always some direction in the spiral. How do you define it? Which is the speed? Nanometers per second, dimensions per second, or is how many terms per second? And if there is a difference.
Yeah, that's a good question. So I define the slow scan axis in the spiral as in radially, because the fast one in a spiral would be kind of tangentially, but the slow one at the end of the scan is radially. And in the flower, where you're going kind of like this, the fast one would be radially, and the slow one would be angular. That makes sense. Now you can start calculating speeds, radial speeds, tangential speeds. This will depend strongly on what spiral you're, patterning. I can do spirals with 4 inner spirals, or spirals with 22 inner spirals, and then the local velocities will be strongly different. So you can match it with the slow scan speed of a raster scan if you want. As I said, when you try to apply this trailing electric field, which if you think there's no trailing electric field. Like, when you scan, the electric field is sort of instantaneous, but it's not, once you're in the top of the image, there's not an electric field applied in the, in the bottom of the image. So, yeah.
This is the question, no? Why this happens is still...
I think, as I said, it has to do with the hierarchy of the domains that are, given by the energy landscape of the material, coupled to the nucleation that you have under the tip, which is where you have a real electric field.
Okay, so one more questions. So we close. One question.
Just a random thought. So, this on-demand patterns to create this topology, but those you apply the electric field to the tip. I'm wondering, have you ever tried that? No apply electric field, but use a harder tip. And
it's on the to-do list. Mechanical scan path dependent writing.
Get something similar.
Yeah, we can talk about it. You can send me samples. We can try to do it. So I know you guys are experts on the mechanical reading and writing, right? So, yeah, we can talk about it.
Okay, so we close. Yeah. Okay. So thank you.
Thank you.